I'm here with Dr. Michel Sadelain, who is the the director of the Center of Cell Engineering at Memorial Sloan Kettering. Doctor, thank you for joining us today.
My pleasure.
Immuno-oncology is receiving a lot of attention of late because it's producing some rather dramatic results. Talk a little bit about your involvement in immuno-oncology and sort of the groundwork that you're doing, and then talk about some of the challenges that the field is still facing.
Okay, sure. Well, indeed, immuno-oncology is going through a wave of enthusiasm. It started just 3-4 years ago, but obviously this has been 20 years in the making. In December of 2013, Science magazine proclaimed cancer immunotherapy to be the breakthrough of the year. And there were two forms of immunotherapy that were highlighted in that editorial. One of them is checkpoint blockade therapy, and that is based on the use of monoclonal antibodies that de-repress, if you like, latent immune responses that many patients have. They actually have an embryonic immune response, if you like, against their tumor, but it's blocked. It's blocked by molecules that put the brakes on the immune system, and you can release those brakes with an antibody therapy, at least in some patients. And the other one is called CAR therapy, and that CAR stands for chimeric antigen receptors, and those are molecules that actually Memorial greatly contributed to develop. Those are synthetic receptors that we can introduce in patients' immune cells, in this way targeting those T cells to recognize and then attack and kill the tumor. And those have produced breakthrough results in leukemias and lymphomas. In fact, a number of checkpoint blockade therapies and CAR therapies have received FDA breakthrough designation in the past 3 years.
Mm-hmm. So we have a biologic approach and a synthetic approach. And are these complementary? Talk about how these might be able to work together.
So, great question. They've been developed separately, by scientists and companies exploring different angles to tackle cancer. But it may well be that they will turn out to be very complementary. The checkpoint blockade therapy releases an immune response which is already there in the patient, and we know that that's more likely to happen in tumors that have a high mutation burden. The more mutations there are, the more it is likely that some of those mutations will create antigens, now often referred to as neoantigens, that the immune system will recognize. However, not all cancers have high mutation burdens. And in those, again, it's a probabilistic argument. It is possible that checkpoint blockade will not work, at least in some of those patients. Again, if there are no T cells, the checkpoint blockade has nothing to release. And in those cancers in particular, we believe that an engineered immune response may fill the gap. Furthermore, there's room for combining the two. Because checkpoint blockade releases the activity of T cells, we see sometimes, certainly in mouse models today, that the CAR T cell can do a good job, but sometimes it too can be inhibited in the tumor microenvironment. And so a combination with checkpoint blockade may synergize with the CAR T cell. So the two really have followed separate routes, but could well be complementary.
What's interesting about the combination opportunities with immuno-oncology is whether or not it will force the industry and academics to work more closely together. You're obviously on the academic side, but you also have a big clinical. You have a lab as well. Talk about how these combinations might actually spur further collaboration.
Well, indeed, combinations are very likely to increase in number, and that is because of the complexity of the regulation of this immune response. It is really wonderful that so many patients already respond to just the blocking of one molecule, the PD-L1 or the PD-1 receptor. Having said that, most patients still do not respond. And so what can we do? Well, there are other checkpoint blockade molecules. Some say there could be as many as 100. There's perhaps still some to discover, but many of these have to be tested in the clinic, and it may be that in some cancer types or in one patient's cancer, this checkpoint blockade or another is more effective. And so this will require combinations. And likewise for the combination with CAR T cells, same principle applies. There are many resistance mechanisms that need to be overcome, but the story of the success of both of these approaches is that they alone already can yield very high-level responses. So sometimes there are hurdles in bringing about these combinations because perhaps the owners of these drugs are different companies that sometimes compete. But what I see is at our center, in particular at Memorial, we've been able to bring together agents from different companies into collaborative studies. And so while there are hurdles of that nature, I think that the pressure to do good for patients and also to move fast in a very competitive field will be compelling to facilitate these collaborations.
Mm-hmm. You know, you just mentioned you just had a panel, and one of the topics that was a part of that panel was personalized medicine. And talk a little bit about how the immuno-oncology space and some of the things that you're doing at the target level in terms of new identification of targets, how that could fit into the broader universe of personalized medicine.
Sure. So the concepts of precision medicine intersect with immunotherapy at many levels. Clearly, the genetic analysis of the tumor through sequencing can reveal some of the antigens, the neoantigens and others that could be found in a tumor, and that could guide a therapy. The analysis of the tumor and its microenvironment using not just sequencing but immunohistochemistry or other techniques can guide the decision-making for which checkpoint blockade to use. Yet another third level is through the systematic analysis of large panels of tumors, we and others are looking for better targets for CAR therapy. So there's no doubt that it's the rule really in the world of cancer at this time that a deeper and closer look at individual tumors is going to guide many therapies in different ways.
Mm-hmm. Very exciting field. Dr. Sadelain, thank you very much for joining us.
It's a pleasure. Thank you.